Okay, we're going to talk about the required practical, which is about photosynthesis. And the aim of this is to find out the effect of changing light intensity on the rate of photosynthesis. Okay, so how are we going to measure the rate of photosynthesis? We look at the equation there. So we've got carbon dioxide plus water gives you glucose and oxygen. Right, so you could measure the rate at which carbon dioxide is used up. That's a possibility. We could measure the rate that you make glucose. That would be quite tricky to do, and the easiest way to do it is to measure the rate of production of oxygen. Okay. Now, if you've got a normal plant, it would be pretty difficult to do that. A land plant, because the oxygen it release makes will just mix with the air. But if you use an aquatic plant, the oxygen that it's made isn't very soluble in water, so it makes bubbles, and you can basically count the number of bubbles being made, and that will give you a rough idea of the rate of photosynthesis. So this is the apparatus which you will use. Right, we've got some pondweed in there, and a couple of bits of pondweed in a beaker of water. Right, and any oxygen that is made is going to rise, and it's going to get trapped by this funnel, this inverted funnel here, and the bubbles of oxygen are going to rise, they'll float, and you can see them, you can count them as they go through the test tube there. And of course what's collecting there is oxygen gas. There's some things where you actually use an inverted measuring cylinder, then you can calculate the volume of oxygen gas made. But the volume is quite small, so it's not that practical. And this is a tricky experiment to get decent results for, it's got to be said. These little red things here, it says to put the filter funnel on 2 pieces of plasticine so that all this water can circulate around. Okay, now let's think what. Okay, of course, I forgot to mention we've got our light here, and we're going to change the light intensity by moving the light closer or further away to the beaker where the photosynthesis is occurring. Okay, so let's think about our variables for the experiment. So the independent variable, the one I am changing, that is the light intensity, or if you like, the distance the light is from the beaker. And the dependent variable is the rate of photosynthesis, which is the number of bubbles counted in 30 seconds. Or you could count it in a minute, whatever. Yeah. Right, now let's think about control variables. What do we need to do to make this a fair test? Well, what else affects the rate of photosynthesis? Well, the temperature is pretty crucial, and so we have to be careful here that the temperature doesn't change, and this light might get quite hot, and that might warm up the water. So you have to be careful that that's not the case, particularly when it's quite close up to the beaker, like here. And what you can do is you can put a piece of glass, thick piece of glass, in between the light and the beaker. The glass will absorb the infrared, which is infrared radiation, which is responsible for making the water get warmer, but it won't stop the light getting through. It's completely transparent to that. So that's what you can do there to control for that problem. Another thing will affect the amount of the rate of photosynthesis will be the CO2 concentration. Well, we're going to presume that the CO2 concentration in this water doesn't change very much because the pondweed is not using up that much CO2, and all the time more CO2 will be absorbed from the atmosphere. Okay, so we're going to just presume that stays constant. The other thing that would affect the rate of photosynthesis, of course, is the amount of chlorophyll. Okay, you've got there, and that is going to stay the same because you're going to just leave the same 2 bits of pondweed in the apparatus for the whole experiment. Okay, so that's things, what you got to think about in order to make it a fair test. Okay, control variables. Okay, so now let's think about how we're going to change the light intensity. And this is a little bit tricky because you see here, light intensity is proportional. This little this sign here is proportional. That means to 1 over the distance squared, not just the distance, 1 over the distance squared, right? And let's see why that is. A little diagram here. Why is that the case? Well, here we've got I've got that yellow dot is supposed to represent the light, and let's say that this red circle, this has got a radius of 1 meter, and the blue circle has got a radius of 2 meters. Now, the light made from the lamp, if you are just 1 meter away, it is spread out over all of that area, that red area there. Now, what's the area of that circle? Well, that's the area of the circle is going to be is equal to pi r squared. So in the case of the red one, it is equal to pi 1 squared, which is just of course 1. Whereas when you're 2 meters away, you've got the same amount of light, but it's spread over a bigger area. Right, now what's the area of the blue one? Right, the area is going to be equal to pi r squared again. That's equal to pi 2 squared, which is equal to 4 pi. And this one, of course, is just equal to 1 pi, or just pi. So what's the ratio of the areas? As you increase, the ratio is 1 pi to 4 pi. In other words, divide through by pi, it's 1 to 4. So by doubling the radius, we have increased the area that the light is spread over by a factor of 4. In other words, 2 squared. And that's where this is why it's proportional to 1 over distance squared. So now let's say we're going to measure the light intensity. Probably best to use it in arbitrary units. So let's say at 10 centimeters, when the light is distance of 10 centimeters, the light intensity is equal to 1,000 units. Okay, let's say that. So how do we work out what is the light intensity at 20 centimeters? Well, the way we do that is, well, we've got light intensity is proportional to 1 over d squared. Now you can always replace the proportional signs by putting a constant in there. So I just call that light intensity. Replace the proportional sign here with an equal sign and a constant. So that's going to be equal to constant, which is little k, multiplied by 1 over d squared. Let's rearrange that to work out what the value of that k is. So k is equal to d squared multiplied by the light intensity. So let's say when the distance is 10 centimeters, we know that the light intensity is equal to 1,000. So the constant there is going to be equal to 10 squared multiplied by the light intensity, 1,000. That is equal to 100,000. So now let's work out what would be the light intensity at, shall we say, 25 centimeters cubed. Okay, so let's do that. So at 25 centimeters cubed, light intensity is equal to constant multiplied by 1 over d squared. So that's equal to 100,000 multiplied by 1 over, well, d squared is 25 squared. So we do that on a calculator. is equal to 160 units. Okay, so what I have done here is this whole table here is I've done the same thing for all of those different distances, and you can see how the light intensity changes as the distance increases, and you can see it drops away very rapidly. So it's 1,000 units when it's 10 centimeters away. 20, it's gone down to a quarter of that value. At 40 centimeters cubed, it's really small. It's only 62 units. It drops off very quickly, and that's because of this 1 over d squared relationship. Right, now let's have a look at the expected results, and you'd probably be quite lucky and very skillful to get a set of results like this from this sort of experiment. Right, so here's rate of photosynthesis here, and here's the light intensity. And it should go up at first, it should be fairly okay, and then it gets to a point when increasing the light intensity, about here, increasing the light intensity doesn't increase the rate of photosynthesis anymore. So we'll cover it in a later video, another video, but we say here that the rate is independent of the light here. Of the light. Or another thing we say is light is not a limiting factor. It's something else which is stopping the rate being higher, not the amount of light, such as the carbon dioxide concentration. If you increase that, you might go faster, or if you had more chlorophyll, put more weed in there, it might go faster. Whereas at this point of the graph here, down here, we would say the light is the limiting factor. You can make the reaction, the photosynthesis, go faster by giving it more light. Okay, we'll cover those things in a later video, but really this one is just to get an idea of what this experiment is about. What are the variables? What are the control variables? What kind of calculations might you be expected to do with it, and the expected conclusions.